you will remember that you're mostly made up of water. But imagine that instead of skin, the only thing that's holding you together is a thin bilayer of lipids or fat components. Also imagine that you're only as wide as 1/5 the diameter of human hair, or about 10 microns. Congratulations, you're a cell. There's only one problem. Surface tension would force you to collapse into a sphere, and cells are rarely spherical. They have many different shapes. Just think about the shape of a neuron, forming structures such as filopodia and lamellipodia, which are projections at the edge of the cell that are filled with actin, and undergo different processes such as cell motility and phagocytosis, where the cell basically engulfs and eats bacteria and other solids. All of these and more are dependent on actin, a protein that is found in all eukaryotic cells and that forms the cell's skeleton, which is much more flexible than our skeleton and much more prone to changing structure. Here are a few microscope images that better illustrate some of the concepts on the previous slide, such as the sheet-like lamellipodia and these stress fibers throughout the cell, which help the cell adhere to a surface. In this movie, you can see cell motility, where there are filopodia and lamellipodia forming at the edge of these cells as they move. With actin playing such an important role in eukaryotic cells, pathogens have evolved so that when they invade cells, they can hijack the cell's actin cytoskeleton to form actin tails, which mediate actin-based motility to allow the cell to move around the cell, or to allow the bacteria to move around the cell and spread to cell to cell while evading the host's immune system. In this diagram, we see a bacterium forming a protrusion in the process of cell-to-cell spread. Burkholderia species are gram-negative intracellular bacteria that can invade cells and form actin tails, but the host's and ability to cause disease by these species widely differ. Hylodensis, which I'll refer to as BT, is generally avirulent. Pseudomallii, or BP, infects people and causes melioidosis, which is endemic in Southeast Asia and Australia. Patients with melioidosis usually have fevers and other symptoms that suggest tuberculosis or pneumonia. Mallii, or BM, infects mostly equines, such as horses, but can be transferred to humans, and causes glanders in Asia, South America, the Middle East, and Africa. Signs of glanders in humans include lesions in the lung, followed by septicemia. These Burkholderia species require a conserved bacterial protein called BimA to form actin tails in cells. BimA is a member of trimeric autotransporters, which basically means that 3 individual BimA proteins trimerize to form a complex together. In this image, the Pseudomallii bacteria, colored in red, is about to divide, and BimA, colored green, localizes to one pole of the bacteria. BimA nucleates actin at one bacterial pole, which initiates the formation of the rest of the tail. And you can see these red bacteria forming green actin tails in this cell. To explain what I mean by nucleate actin, we first need to take a step back to see how actin works in the cell. Actin exists in 2 forms in the cell, as individual monomers, or G-actin, and as filaments, or F-actin. To go from G-actin to F-actin, 3 actin monomers were combined to form a C in a slow process called nucleation, and subsequent addition of actin monomers rapidly polymerizes the rest of the tail, or the filament. Nucleation is the slowest step in actin polymerization and is highly regulated by the cell to allow rapid filament formation only when it's required. Cells use a number of different proteins to nucleate actin, and these actin nucleators include the ARP2/3 complex and tandem nucleators. The ARP2/3 complex is bound by a nucleation promoting factor, or NPF, to initiate a new filament off the side of an existing filament. Tandem nucleators nucleate filaments by binding G-actin monomers to form the nucleation seed. The sequences of BimA from BT, BP, and BM suggest that they mimic cellular actin nucleators, but that the mechanism of actin nucleation by these BimAs differ. BT BimA contains a WCA domain, which consists of a predicted WH2 motif that binds monomeric actin that is adjacent to a central and acidic domain. The WCA domain is found in cellular NPFs that activate 3, which suggests that BT BimA also binds ARP2/3 to nucleate actin. BP doesn't have a WCA domain and instead has 3 WH2 motifs in tandem, which suggests that it may bind and nucleate actin in a manner that is similar to the cellular tandem nucleators. BimA only has 1 WH2 motif, but because BimA proteins are trimeric, 3 total WH2 motifs would bind and nucleate actin like a cellular tandem nucleator. These unlabeled boxes indicate other sequence differences that may also have an effect on actin assembly, but today I'll just focus on the domains that I've already talked about. These BimA sequence differences suggest different mechanisms of actin nucleation, and the goal of this project is to determine how the mechanism of actin nucleation by these BimAs differ. To elucidate these predicted differences, we first need to answer if the BimA proteins alone are sufficient to nucleate actin in vitro, and then we can begin to identify the specific motifs that are required for BimAs to nucleate actin and mediate actin-based motility. Most of my work this summer has been toward answering the first question. I've expressed the 3 BimAs, figured out purification schemes for each of them, and assayed for activity by mixing together G-actin monomers and a purified BimA using a pyrene-actin polymerization assay. In this assay, actin monomers that are labeled with pyrene will fluoresce when they're added to a filament. And that fluorescence can be measured over time. The rate of fluorescence will be greater if these BimAs are active and nucleate actin, because more filaments would form relative to spontaneous actin nucleation and polymerization, meaning that more pyrene actin would be incorporated into filaments. I used the pyrene assay to test if the BimA constructs that I purified were active and if they were sufficient to nucleate actin by themselves, and if not, did they nucleate actin in a manner that was dependent on ARP23. Based on the WCA domain and VT sequence, we predict that it would require ARP23 to nucleate actin. Looking at the data, VT does nucleate actin in a concentration-dependent manner, as represented by this gradient. And this black line shows the rate of actin polymerization by spontaneous actin nucleation, whereas you see the orange curves shifting toward the left, which indicates increasing activity with increasing VT concentration. This suggests that VT works as a nucleation promoting factor that binds ARP23, but what was interesting was that it could also nucleate actin independently of ARP23, where you can see the same pattern of activity, but in the absence of ARP23, this activity is much more modest. We only start seeing a noticeable difference at higher concentrations of VT. Because VT is a trimer, this suggests that the 3 total WH motifs can nucleate actin like a tandem nucleator, but that the predominant mechanism of actin nucleation is that of a nucleation promoting factor that binds ARP23. And it raises the question if BT or thailandensis can actually use both mechanisms during a live cell infection. So yes, we can say in the context of this assay that BT is active, but that it requires ARP23 for maximum actin nucleation activity. In contrast, we predict that BP nucleates actin in a manner that is independent of ARP23. In the absence of ARP23, BP nucleates actin in a concentration-dependent manner as well, and we also see an effect on the steady-state levels of pyrene fluorescence after the initial spike in polymerization. So in this case, not only did the curve shift left with increasing BP concentration, but they also shift up. Other sequences in BimA may be causing this effect on the steady state levels. The pattern of activity is the same in the presence of ARP23, which gives evidence that its predicted tandem nucleation mechanism is independent of ARP23. And then, so we can say that this BP construct is active and nucleates actin independently of ARP23. And finally, given that BM has 1 WH2 motif and that it trimerizes, we predict that its mechanism of actin nucleation is also independent of ARP23. So like BP, BM can nucleate actin in a concentration-dependent manner, and it also has a similar effect on the steady-state levels. This activity remains the same in the presence of ARP23, suggesting that its mechanism is also similar to tandem nucleators. And again, we can say that BM is active and doesn't require ARP23, which is what we predict if BM also uses the tandem nucleation mechanism. In summary, All three BimA constructs can nucleate actin, but BT alone is not sufficient and requires ARP2/3 for maximum actin nucleation activity. And going back to the 3 actin nucleation models, BT seems to mimic cellular tandem nucleators, cellular nucleation promoting factors that bind ARP2/3 to nucleate filaments, while BP and BM are more similar to the tandem nucleators. And here are models of how each Burkholderia species may use BimA to nucleate actin. We can see that BimA trimerizes at one bacterial pole of these red bacteria, and in BT's case, the WH2 motifs bind monomeric actin, and the CA domain bind ARP2/3 to nucleate a new filament in a branching pattern. For BP and BM, the WH2 motifs bind actin monomers to form the nucleation seed to nucleate new filaments. Although we have a better understanding of the mechanism of BimA actin nucleation, we can further characterize the effect of each BimA on motility. We can test this effect in vitro by using a bead motility assay, by coating microscopic beads with BimA, adding the beads to cell extract, and then using time-lapse microscopy to image motility. I'm also working on generating BimA constructs that have specific mutations to the actin-related sequence motifs to identify which of those motifs are required for actin nucleation. I'll express and purify these mutant BimAs in an assay for activity using the same in vitro assays I've already described, and I can also generate tylodensis strains that express these mutant BimAs to see the effect on tail formation and motility during a live cell infection, and measure the efficiency of cell-to-cell spread in a plaque assay. And finally, I'd like to thank the many people without whom this project really wouldn't be where it is today, especially Matt Welch and Aaron Benanti for their endless patience and guidance, the rest of their Welch lab for their support, and of course the Berkeley CERF program, the Pergo Fund for giving me the opportunity and the monetary support, and Leah Carroll and Nathan Shea for their excellent advice and helping me develop this project over the summer. Thank you.